Hello everyone, welcome to our next video, and this is going to be the third of the 3 lectures of types of amino acids, and we are going to deal with 5 amino acids which have polarity as well as charge. And out of those 3 amino acids we have out of those 5 amino acids we have 3 amino acids which will be having positive charge and 2 amino acids which will be having negative charge. So let's get into those without wasting much time. Now let us see the 3 positively charged amino acids which are also polar. Okay, so let's get started with those. The first is lysine, Lysine, lysine. The lysine has a long R group, long chain, where it contains CH2, 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 4 carbons and 1 amino group. This amino group here is just like this amino group out here, and it has a positive charge. This charge gives it the polarity. Since it has a positive charge, so it has one positive pole created towards this, and hence this becomes polar, and this will react with water. Polar compounds always react with water because they have a charge. So in the body, this lysine can be reactive to water. Okay. So here we have one amino group. So lysine contains an amino group. You should remember that lysine contains an amino group. If we move to arginine, here this is the side chain, another huge side chain, long one. Here we have 3 carbons, then a nitrogen, then another carbon, and again amino group. But this group, this C double bond NH2, this sort of groups are called guanidium groups. Okay, this is not an amino group. This C double bond NH2 group is called guanidium. So arginine contains a guanidium group which gives it the polarity. This positive side will give it the polarity. Okay, I hope that is clear. In lysine it is NH3 plus, which is the polar part, amino group, and here we have a guanidium group that gives arginine the positive charge, making it a polar hydrocarbon. Polar amino acid. Moving into the third one, we have histidine. Histidine contains this 5-membered ring called the imidazole ring, and this is our R group. This imidazole ring is an aromatic ring, obviously, and also this N here has the positive charge because of double bond, single bond, and one hydrogen attached. So we have one positive charge with this, and this gives this molecule the polarity. Now there is one catch with this histidine. All these have positive charge. Both these have positive charge at neutral or physiological pH. That is the pH that is maintained in our body for proper functioning. Histidine, although we have shown that it has positive charge, this histidine can also have a neutral charge depending upon the local environment. If the environment in which it is present supports its positive charge, then it will have a positive charge, or it will lose this hydrogen, become a neutral atom. So histidine can be both neutral or positive at physiological pH depending upon the local environment. Okay, I hope that is clear. Based on the local environment, this can become either positive or neutral. So here are the 3 positively charged polar amino acids, lysine, arginine, and histidine. Very simple. Remember the groups. Here we have one amino group, here one guanidium group, and here we have an imidazole ring in which one of the nitrogen is positively charged at physiological pH. So that is clear. Now we will move into the remaining 2 negatively charged amino acids. Now we are into the negatively charged amino acids. The first one is the aspartate or aspartic acid. The second one is glutamate or glutamic acid. So we have 2 only. So in aspartic acid, this is our side chain, a very small side chain, CH2 COO minus. So we have this carboxyl group giving it the negative charge and hence the polarity. Then we have the glutamate or glutamic acid. In this case, this is our side chain, only one extra CH2 molecule attached in compared to the aspartic acid or aspartate. Here we have one CH2COO, we have CH2CH2COO minus. So this gives the negative, this carboxyl group gives the negative charge to glutamic acid, making it a negatively charged amino acid. So these are the last 2 of the amino acids that we will study in the standard amino acids. We have two more, which are selenocysteine and pyrrolysine. Now, as I mentioned earlier in one of my videos, that pyrrolysine and selenocysteine are actually derivatives of these 20 amino acids that we have studied. Till now, we have studied 20. In the uncharged non-polar amino acids, we had 9 of those. In uncharged polar amino acids, we had 6. And in today, now that we have studied in polar amino acids, charged polar amino acids, we had 5. So 9 plus 6 plus 5 gives 20. So 22 amino acids are there. So two more are left, which are selenocysteine and pyrrolysine. And those are derivatives of these 20 amino acids. So we'll study about this in our next video, because we also have some non-standard amino acids to talk about. So without making this video unnecessarily long, I will take those two topics in my next video, which is going to be on pyrrolysine, selenocysteine, and also some of the non-standard amino acids. As we all know, as I said earlier, there are 300 amino acids in total known till date, out of which only 22 are found in the proteins. So remaining 278 amino acids, what are their functions? Obviously, we are not going to study about 278 amino acids. We will focus on a few important ones, and we will see what are their uses, so that we can get an idea of what non-standard amino acids actually do in our bodies. Okay, so I hope that is clear. A very short video today, not that lengthy. So if you have any confusion regarding this, do ask in the comment section. You can follow me on Facebook at Tirthankar Roy, and also at Instagram at Tirthankar Roy. So do follow me there. You will get some insights to some other topics as well. So hope you liked it, and until next time, peace.